My name is Kent Lloyd, and I am professor in the Department of Surgery in the School of Medicine. I lead the UC Davis Mouse Biology Program in the Office of Research, and I also serve as Associate Director of Shared Resources for the Comprehensive Cancer Center. "25 years ago, we decoded the human genome and identified about 20,000 protein-coding genes. Now we know that genes encode proteins that play biological roles in things like how tall you are and your eye color, and pathological roles in diseases like heart disease, Alzheimer's, and cancer. But back then, we didn't know what most of these 20,000 genes do. So to find out, we've been trying to connect the dots between genes, the proteins they encode, and the diseases that they play a role in. So for the last 20 years, I've been leading projects and collaborating with scientists across the US and around the world to understand what all these genes do in both health and disease. To understand gene function, my research uses mice and mouse stem cells because they have nearly all the same genes as in people. By using a complex biological system like a mouse, my research enables me to decipher what these genes do so we can better diagnose, treat, and prevent disease. And this work is essential for validating novel alternative methods that don't use animals. Here at UC Davis, we've led research that has already discovered the function of about 2,000 genes, which is nearly 10% of the genome. We've discovered new mechanisms that have identified drug targets for heart disease, schizophrenia, depression, diabetes, deafness, and many other diseases and conditions. And as a veterinarian myself, my work can also impact our ability to diagnose, treat, and prevent diseases in pets and domestic and wild animals. The return on NIH investment in my research to date has far exceeded our expectations. And this is what science is all about, the ability to discover new knowledge that we didn't know or couldn't do before. It's giving us new clinical insights into new drug targets, new uses for existing medications, and validating prevention strategies. Now, as everyone knows, at this very moment, federal funding for life-saving biomedical research is under threat. Just imagine, if you will, if funding for the Human Genome Project had been cut halfway through the project. If that funding had been cut, so many medical breakthroughs of the past 20 years would just not have happened. There remain many thousands of genes for which we still don't know what they do. We haven't finished our work in this area, but we're making great progress. So if our funding were cut back, we would be left in the dark about the function of thousands of genes. It would set back our understanding of human biology and the discovery of new diagnostic tools and treatments. We stop federal funding for this kind of research at our peril.